In this video, we will discuss about protein translocation, the types of protein translocation, and there are three types of protein translocation. First one is co-translation translocation. Second one is post-translation translocation. Third one is post-translation translocation. However, the difference between second and third is that third one occurs in the eubacteria, second one occurs in yeast mammals, and first one occurs in also occurs in mammals yeast. So we will discuss just a brief idea about the different types, and in subsequent video we will discuss in details about each type. So this co-translation translocation is the general process that we have already touched upon this during end glycosylation pathway. This is the general process. This is the general process. Okay. So what happens is that those proteins that are destined to move to the membrane, that is inside ER lumen, has SRP signal, that is as SRP sequence. SRP is signal recognition particle. So whenever this SRP sequence is synthesized, the ribosome machinery stops further sequencing of the mRNA and transport the whole machinery to the ER membrane, where it forms a complex that is we have discussed SEC 61, and that helps in and as soon as they forms a complex with the membrane, the translation process again begins, and the polypeptide chains begin to grow inside the ER lumen. So this is the general process, and how this occurs in detail we will discuss it in the next subsequent videos. And in this protein translation translocation, what happens is that the hydrophobic SRP sequence, that is the hydrophobic sequence, is small. Hydrophobic sequence is small. So because of this, the SRP sequence not able to interact with SRP receptor or SRP membrane receptors. So in this case, the whole translation occurs in the cytosol, and the translocation process occurs with different complex that is been named as SEC 62-63 complex, and this complex works in coordination with BIP, that we have also discussed BIP during N-glycosylation process. BIP is a member of HSP70 protein. HSP70 protein. That also helps in proteins to get their native structure. So this complex SEC62-63 complex helps the protein that are translated in the cytosol to translocate it further to the final destination, or either in ER lumen. From there, the further post-translation modification occurs, then they move to the final destination. And this protein and this again post-translation translocation in eubacteria, that is usually occurs with the help of cytosolic ATPase. And this is another name for this is the SEC-A. So it forms whenever a polypeptide chain comes in contact with SEC-A, there is a conformation change occurs. and this conformation change lead to further interact with cytosolic cycle. Cytosolic cycle helps to interact with cytosolic cycle that helps in that helps in translocation of protein after translation, and this is mainly for the secreted proteins, that is the proteins that are destined to move outside the cell or in extracellular matrix. So this is just a general idea about how the protein translocation process is characterized in different process. How protein translocation is characterized based on the different processes. So that's all for this video. Thank you for your time.